Welcome to the video on radiobiology, where we delve into the fascinating realm of how radiation impacts biological systems. From understanding the fundamental principles of radiation interaction with living organisms to exploring its implications on human health, this video aims to provide a comprehensive overview of the effects of radiation on biological systems. Hello everyone, I'm Dr. Tanushree. Get ready to uncover the complexities of radiobiology and its significance in shaping our understanding of the intricate relationship between radiation and life. What is radiobiology? Radiobiology, also known as radiation biology. It's a branch of science that deals with action of ionizing radiations on biological tissues and living organisms. It's a combination of two disciplines, radiation physics and biology. Now, what is ionizing radiation? Ionizing radiation consists of subatomic particles or electromagnetic waves that have sufficient energy to ionize atoms, molecules by detaching electrons from them. So, energy of ionizing radiation starts between 10 electron volts and 33 electron volts. So, as shown in this figure, different types of electromagnetic radiations. So, gamma rays, X-rays, and the higher energy ultraviolet rays are part of electromagnetic spectrum are ionizing radiations. However, the lower energy ultraviolet, visible light, nearly all types of laser light, infrared, microwave, radio waves are non-ionizing radiations. Typical ionizing subatomic particles include alpha particle, beta particle, and neutrons. These are typically created by radioactive decay, and almost all are energetic enough to ionize. So let's know about these subatomic ionizing radiations in detail. Isotopes of elements with high atomic number frequently decay by emitting alpha particle. So radioisotopes, some of the radioisotopes decay by emitting alpha particle. So what is alpha particle? Alpha particle is an helium nucleus. It consists of two protons, which are indicated with the pink balls, and two neutrons, which are indicated by the blue balls. So they are bound together. Two protons and two neutrons are bound together into a particle identical to a helium nucleus. Therefore, alpha particle is also known as helium nucleus. Now, alpha particle emissions are generally produced in the process of alpha decay. Emission of alpha particle result in considerable lightening of the nucleus, a decrease in atomic number of 2, and decrease in mass number of 4. So basically in alpha particle emission, in the radioisotopes which undergo decay by emitting alpha particle emission, the loss of 2 protons and loss of 2 neutrons occur, that is the helium nuclei is emitted, alpha particle is emitted. Now number of proton is nothing but the atomic number. So this is the reason I am saying that emission of alpha particle results into lightening of the nucleus with decrease in atomic number of 2, and because mass number is given by sum of atomic number and number of neutrons, so mass number decreases by 4. So as shown over here, the example is of uranium-238. So here the protons are indicated with blue balls and neutrons are indicated with the pink one. So when the alpha particle is emitted, you can see that there is a loss of 2 protons. So from uranium, if 2 protons are emitted, that means there is a decrease in atomic number by 2 and of the daughter nucleus. In the periodic table, the element having the atomic number 90 is thorium. So uranium is converted into thorium, and because the mass number decreases by 4, So 238 is the mass of the parent nucleus, and because there is a mass number decrease of mass number by 4, so the mass number of the daughter nuclei will be 234 as shown over here. Now if this thorium is unstable, its nuclei is unstable, then it will further undergo decay by emitting the subatomic particles, alpha particle, beta or gamma rays. Now kindly note that isotopes that decay by alpha emission are not frequently encountered in biological work, although they can be found in instruments such as scintillation counters and smoke alarms. Then beta particles are high energy, high speed electrons or positrons emitted by a certain type of radioactive nuclei like potassium 40, and some more examples I have quoted here. So the production of beta particle is termed beta decay, and it is designated with a Greek letter beta as shown over here. Now there are two forms of beta decay, beta minus and beta plus, which respectively give rise to electron, that is negatron, and the positron. So let's know about beta minus decay in detail. In beta minus decay, a neutron is converted to proton by ejection of a negatively charged beta particle called as negatron. To kindly note, to all intents and purposes, a negatron is an electron only. But the term negatron is preferred since it serves to emphasize the nuclear origin of the particle. So as a result of negatron emission, the nucleus loses a neutron but gains a proton. Therefore, atomic number increases by 1 and mass number remains constant. For example, carbon 14, it has 6 protons and 8 neutrons. Due to unequal number of protons and neutrons in the nuclei of carbon 14 is unstable, and hence carbon 14 is radioactive. That is, it regains stability by emitting the radiations from the nucleus. Now here the kind of decay it undergoes is beta minus decay, or beta particle is emitted, that is negatively charged beta particle is emitted, called as negatron. So as I told you, to all intents and purposes, negatron is electron only, but because it has the nuclear origin, the term negatron is preferred, and along with it some particles are also released like antineutrino. Now since the neutron, like carbon 14 had initially 8 neutron, now 7 neutron remain as it is, and one of the neutron converted into a proton. So because of one neutron converted into a proton, the final number of protons becomes 7 and neutrons 7. So initially 6 plus 8, 14, that is the mass number, and after emission also 7 plus 7, 14 is the mass number. So mass number doesn't change because interconversion is happening, neutron is converted to proton. But atomic number increases by 1. So you can see that parent nucleus carbon 14 is converted to nitrogen 14. Negatron emission is very important to biochemists because many of the commonly used radionuclides they decay by these mechanisms, like for example tritium, carbon 14 as I showed you. So they are used to label organic compounds. So in carbon dating we use carbon 14 to determine the age of the fossils. And then even in molecular studies, in order to study the proteins, we use 35S, which is used to label methionine to study protein synthesis. Then 32P can be used to study nucleic acid. So it's a very powerful tool in molecular biology when used as nucleic acid labels. Then in beta plus decay, opposite happens. Just opposite of beta minus decay happens. Like here, proton is converted to neutron. For example, carbon 10, it has 6 proton and 4 neutrons. So here 5 protons remain as it is and one of the proton is converted to neutron. So the total number of neutrons now becomes 5 neutron. And because one of the proton is converted to neutron, the final number of protons now left is 5. So atomic number, that means decreases by 1. And mass number, so initially 6 plus 4, 10 is the mass number. But even after this proton converted to neutron, because interconversion is happening, there is no loss of as such proton or neutron happening here. So mass number remains same, that is 10. But the parent nuclei, that is carbon 10, is converted to boron 10. So here the nucleus emits a positron, which is a positively which is a positive particle like an ordinary electron in mass but of opposite charge. Radiative photons are called as gamma rays if they are produced by nuclear reaction, subatomic particle decay or radioactive decay within the nucleus. However, they are called as X-rays if produced outside the nucleus. So the generic term photon is used to describe both. So as shown over here, plutonium, which has an atomic number of 94 and mass number 240, when its nuclei changes its energy state from higher to lower, it releases photons of light which lies in gamma region of electromagnetic spectrum. So gamma rays are emitted when the plutonium nucleus changes its energy state from higher to lower. Then in some cases, alpha and beta particle emission also give rise to X-rays. For example, iodine 53, so it undergoes beta minus decay. So here the change in atomic number is because of beta minus decay, and along with it you can see gamma radiations are also emitted. Then neutrons. Neutrons are neutral particles. They have neutral electric charge and they are often misunderstood as zero electrical charge, and thus often do not directly cause ionization in a single step or interaction with matter. However, fast neutrons will interact with the protons in hydrogen while linear energy transfer, energy that a particle transfers to the material it is moving through. So this mechanism scatters the nuclei of the material in the target area, causing direct ionization of hydrogen atoms. And when neutrons strike the hydrogen nuclei, proton radiation results, that is fast protons results, and these protons are themselves ionizing because they are of higher energy and are charged and interact with electrons in matter. So if we compare alpha, beta and gamma particles, alpha particles are strongly ionizing form of radiation. But when emitted by radioactive decay, they have low penetration power and can be absorbed by a few centimeters of air or by the top layer of human skin. Then beta particles, they are less penetrating than gamma radiation but more penetrating than alpha particles. However, high energy beta particles may produce X-rays known as Brahmstrahlen, which means breaking radiation, and even produce secondary electrons, that is delta rays, as they pass through matter. And both of these can cause an indirect ionization effect. Then gamma rays, they have low ionizing power but high penetrating power. Now let's understand what happens when cells are exposed to ionizing radiation. When the cells are exposed to ionizing radiation, the standard physical effects between radiation and atoms or molecules of cells occur first, and the possible biological damage to cell functions follows later. The biological effect of radiation results mainly from damage to the DNA, which is the most critical target within the cell. However, there are also other sites in the cell which, when damaged, may lead to cell death. So when directly ionizing radiation is absorbed in biological material, the damage to cell may occur in one of two ways: direct action or indirect action. In direct action, as shown over here, the radiation interacts directly with the critical target in the cell. So here, let's say the critical target in the cell is DNA. So ionizing radiation, they can directly interact with DNA molecule, causing various type of damages such as single strand break, double strand break, base and sugar damage, even cross-linking of DNA strands. So these type of damage can lead to mutations, chromosomal aberrations, and ultimately cell death. However, in indirect action, the radiation interact with other molecules and atoms, mainly water, since 80% of cell is composed of water within the cell to produce free radicals that can through diffusion in the cell damage the critical target within the cell. So indirectly, critical target may be DNA, which can be damaged. So an interaction of radiation, ionizing radiation with water, short-lived yet extremely reactive free radicals such as water ion and hydroxyl radical are produced. The free radical in turn can cause damage to the target within the cell, like as I told you, they interact with the DNA causing mutations. Also, they can interact with the proteins, lipids, leading to oxidative damage and cellular dysfunction. So as shown in this figure, ionizing radiation causes the single strand and double strand breaks of DNA either directly or indirectly, resulting in oxidized proteins, DNA, lipids and other biomolecules. And within minutes, the cell responds by altering gene expression and modifying stress-related proteins. And high dose of radiation leads to cell death or genomic instability. Now, when the ionizing radiation it falls on the target, the time scale involved between breakage of chemical bonds and biological effect depends on type of damage. If cell kill is the result, it may happen in hours to days when the damaged cell attempts to divide. So this is an early effect of radiation. But if the damage is oncogenic, that is cancer induction, then its expression may be delayed for years. So this is the late effect of radiation. So ionizing radiations has been proven to cause leukemia and has been implicated in development of many other cancers in tissues such as bone, lung, skin, thyroid and breast. In addition to carcinogenesis, that is induction of cancer, the late effect of radiation includes lifespan shortening, genetic damage and potential effects to the fetus. Then if the damage is a mutation in germ cell, then the effect may not be expressed for generations. So the fate of irradiated cell, that is irradiation of a cell, will result in several outcomes. First is no effect. Second is division delay, that is the cell is delayed from going through division. Third, apoptosis, that is the cell dies before it can divide or afterwards by fragmentation into smaller bodies which are taken up by neighboring cells. And fourth is reproductive failure, that is the cell dies when attempting the first or subsequent mitosis. Kindly note that cells have evolved various mechanisms to respond to radiation-induced damage. These include DNA repair pathways such as base excision repair, nucleotide excision repair and homologous recombination. Additionally, cells can activate signaling pathways such as p53 pathway to arrest the cell cycle and facilitate DNA repair, or induce apoptosis if the damage is irreparable. Then even we can classify the radiation damage to mammalian cells into three categories. Lethal damage, which is irreversible, irreparable and leads to cell death. Second, sublethal damage, which can be repaired in hours unless additional sublethal damage is added and eventually leads to lethal damage. And third is potentially lethal damage, which can be manipulated by repair when cells are allowed to remain in a non-dividing state. Now let's have a look at the effect of radiation on the human population. The effect of radiation on the human population can be classified as either somatic or genetic. Somatic effects are harm that exposed individuals suffer during their lifetime, such as radiation-induced cancer, that is carcinogenesis, sterility, opacification of the eyelids, and life shortening. Carcinogenesis expresses itself as a late somatic effect in the form of acute or chronic myeloid leukemia or some solid tumors, for example in skin, bone, lung, thyroid or breast. However, genetic or hereditary effects are radiation-induced mutations to an individual genes and DNA that can contribute to the birth of defective descendants. Then stochastic and deterministic, that is non-stochastic effect. The harmful effect of radiation may be classified into two general categories, that is stochastic and non-stochastic. A stochastic effect is one in which the probability of occurrence increases with increasing dose. But the severity in affected individual does not depend on the dose, that is induction of cancer, that is radiation carcinogenesis. Genetic effects. So there is no threshold dose for effects that are truly stochastic. But a deterministic effect is one which increases in severity with increasing dose, usually above a threshold dose in affected individuals, like decrease in sperm count, lens opacification, organ atrophy, fibrosis. Then third type of effect is acute or chronic effect. An organ or tissue expresses response to radiation damage either as an acute or as late or chronic effect. So acute effects, they manifest themselves soon after exposure to radiation and are characterized by inflammation, edema or hemorrhage. Whereas chronic effects are delayed and are characterized by fibrosis, atrophy, ulceration or obstruction of the intestine. Now let's know about total body radiation response and measurement quantities. When a radioisotope undergoes decay, it emits radiations, and the unit to measure radioactivity is becquerel. So becquerel is the SI unit of activity, radioactivity, where 1 becquerel is 1 decay per second. So when the radioisotope undergoes decay, it emits alpha particle or beta particle or gamma rays. So it undergoes decay. A radioisotope undergo decay by emission of alpha particle, beta particle or gamma rays. So these ionizing radiation strength from a point source decreases with the square of distance it travels. And even the intervening medium can also absorb and scatter radiation. Then both dose and counts are used to detect the radiation, depending on the application and the radiation type. So physical dose is measured in grays and biological dose is measured in sieverts. So response of an organism to acute total body radiation exposure is influenced by the combined response to radiation of all organs constituting the organism. So depending on the actual total body dose above 1 gray, the response is described as specific radiation syndrome. So if 1 gray is less than dose, which is less than 10 gray, then it is a bone marrow syndrome. If 10 gray is less than dose, which is less than 100 gray, that is gastrointestinal syndrome. And if dose is more than 100 gray, then it is central nervous system syndrome. So toxicity of radiation is dependent not simply on the amount present, but on the amount absorbed by the body, the energy of the absorbed radiation and its biological effect. So a key aspect of determining toxicity is to know how much energy might be absorbed, just as too much sun gives you sunburn and potentially skin cancer. So higher the energy of the radiation, greater is the potential hazard. So the gray and SI unit is the unit used to describe this. So 1 gray is an absorption of 1 joule per kilogram of absorber. So gray is a useful unit, but it still does not adequately describe the hazard to living organism. So the unit of absorbed dose which takes into account this weighting factor is the sievert. And is known as the equivalent dose. So for beta radiation, gray and sievert are same, but for alpha radiation, 1 gray is 20 sievert. In other words, alpha radiation is 20 times as toxic to humans as X-rays for the same energy absorbed. So clearly, from the point of view of safety, it is advisable to use radioisotopes with lower energy wherever possible. Even the absorbed dose from known sources can be calculated from knowledge of the rate of decay of the source, the energy of radiation, the penetrating power of radiation, and the distance between the source and the laboratory worker. So the rate at which dose is delivered is referred to as dose rate, which is expressed in sievert per hour. And it can be used to calculate your total dose. For example, a source may be delivering 10 millisievert per hour. So if you work with the source for 6 hours, your total dose will be 60 millisievert. Now, the effect of radiation on tissues depends on factors such as type of radiation, dose rate, and tissue sensitivity. So, acute exposure to high doses of radiation can cause immediate tissue damage, leading to symptoms such as radiation sickness. Chronic exposure to low doses of radiation may result in long-term effects such as cancer and genetic mutations. So, the effect of radiation on tissue as a function of dose is measured with assays, and the measurement results are given in form of cell survival curves or dose response curves. So 3 categories of tissue assays are in use. One is clonogenic assays, which measures the reproductive integrity of clonogenic stem cells in tissue, and measurement results in cell survival curves. Second is functional assays, which measures the functional endpoints for various tissues and produce dose response curves, where response is measured on a graded reaction scale or expressed as a proportion of cases where reactions are greater than specified level. And third is lethality assays, which quantify the number of animal deaths after irradiation of specific organ with a given dose. So we have learned so far that what is the effect of radiation on cells and tissues and how can we measure them, or the assays that we can use for measurement of radiation damage. Now, there are various chemical agents also which may alter the cell response to ionizing radiation, either reducing or enhancing the cell response. So, chemical agents that reduce cell response to radiation are radioprotectors. They generally influence the indirect effect of radiation by scavenging the production of free radical. Then there are chemical agents that enhance the cell response to radiation are called as radiosensitizers, which generally promote both the direct and indirect effect of radiations. Examples are halogenated pyrimidines that intercalate between the DNA strands and inhibit repair, and hypoxic cell radiosensitizers which act like oxygen. Then another type of radiosensitizers are compounds containing boron that enhances the effect of thermal neutron radiation therapy. Now, it's not that the radiations have the harmful effect only. Even controlled doses of radiations are used for medical imaging and radiotherapy. So most commonly, ionizing radiations are used for treatment for cancer. So ionizing radiation is used to kill cancer cells or inhibit their growth. So radiation oncologists, they have to carefully plan the treatment regimes to deliver precise doses of radiation to tumor tissues while minimizing damage to surrounding healthy tissues. So controlled doses are used for medical imaging and radiotherapy. However, kindly note, high doses can cause visually dramatic radiation burns or rapid fatality through acute radiation syndrome. Now, since working with radiations is harmful, so safety measures and precautions need to be taken while working with the radiations or radioactive substances. So understanding the effect of radiation on biological system is essential for developing radiation protection guideline and safety measure for workers in radiation related industries, as well as for ensuring the safety of the general population in case of nuclear accidents or radiation emergencies. And even the researchers who are working in the laboratories with the radioactive substances, they need to follow proper safety measures and need to take proper precautions. So while handling the radioisotopes which emit the ionizing radiations, it's very much important that one should wear protective clothing, gloves and glasses. And it's better to use the smallest amount of radioisotope as possible. And even the radioactive materials have to be kept safe, secure and well labeled. Then even the researchers who are working with the radioisotopes or working with the substances which emit the ionizing radiations, they have to work in a defined areas and spill tray. They have to monitor their working area frequently. There should be no drinking or eating allowed in the laboratory. The workers should wash and monitor hands after work is done. And also it is very much important that all local rules like dispensing of the radioactive waste, rules regarding that should be followed. And also, especially when working with penetrating radiations like 32P, that is those radioisotopes and those radioisotopes which emit gamma rays. So maximize distance. It is important that the researchers should maximize the distance between the source and himself or herself, and also minimize the time of exposure and maintain shielding. And also no aerosols, radioactive aerosols. One has to make sure that radioactive aerosols are not created while working with them. Now, in the CSIR NET Life Science exam, candidates may be tested on their understanding of these concepts which we have learned in this video, including the molecular mechanisms of radiation-induced damage, cellular response to radiation, and the applications of radiobiology in medicine and other fields. So here is a question which has been asked in previous year CSIR NET Life Science exam. So like here you can see it's a match the following type question where a question is about the radioisotopes, their half-life is given, that is the time taken by the radioisotope to reduce its activity to half, and the type of particles emitted by the radioisotope. So if you know about the time taken by the radioisotopes to reduce their radioactivity to half from the initial value and the type of particles emitted, you can easily score in such type of questions. So as I mentioned earlier in this video that carbon-14 is used in carbon dating and to determine the age of fossils, and it has a half-life of around 5,730 years. So and also it's a beta emitter. So that means B should be 1 and X, which is only given in option B, and therefore it is the answer. So now as you all are familiar with the radiobiology, you are familiar with what is the effect of radiation on biological system, and you have also learned about the type of radiations emitted by the radioisotopes. So kindly prepare this topic accordingly, because this will help you in solving questions based on this topic in the exam. Thank you and take care.